Caroline said, I will be talking to you about the Flu A/B RSV assay on the Panther Fusion. And I'm also going to talk to you about some of my other studies regarding matrix gene mutations and how these mutations have affected the performance of various PCR assays. And hopefully by combining these two topics, that'll help to convince you how truly I feel anyways, and hopefully you will believe me too, that the performance of the flu detection is quite astonishing on the Panther Fusion. Okay, so my conflicts of interest. The only one that's really related to these studies, though, are that some of these previous studies that I'll be talking about were funded by Roche Diagnostics, and then for this particular study, Hologic paid my travel expenses. Okay, so quick review. Influenza, I'm going to go through some of these slides kind of quickly in the beginning, just in interest of time. But influenza is an orthomyxovirus, 100 nanometer envelope virus, and it's segmented single-stranded RNA with 8 separate segments. There are 3 types of influenza, A, B, and C. And influenza A's are categorized by the hemagglutinin and neuraminidase antigens. And actually, after listening to this morning's talk, there are actually 18 HA types and 11 NA types because there's 2 new viruses discovered in bats. But within the influenza viruses, there's considerable genetic diversity. And what is the reason for this genetic diversity? Well, first is the antigenic drift that can or does always occur. Influenza is an RNA virus, so it uses an RNA polymerase for replication, and RNA polymerases do not have proofreading function. So they are constantly introducing errors when they're replicating the nucleic acid. And on average, there's 1 mutation per virion that's introduced. And the majority of these mutations are going to be deleterious, and you have defective viruses produced. But you're going to have some that are able to replicate, some that are going to have an advantage to replication in, like, let's say, within an adverse immune response to a particular antigen. If it can get by that, that's going to give it an advantage. And then there's the antigenic shift, which it's only seen in influenza A, because only influenza A infects other mammals besides humans. And this is with major changes in the viral genome due to reassortment of the viral gene segments, and particularly with reassortments between animal and human viruses, so that now there's a new virus that humans had not seen before and would not have had an immune response, so that they can then cause worldwide pandemics. Okay, so this slide was provided to me by Dave Wentworth, from the CDC, Chief of Virology, Surveillance and Diagnostic Branch. And it is a phylogenetic tree developed by the NextFlu team. And it's phylogenetic tree showing the viruses over, these are all H3N2 viruses over a 10-year period, and how that they have diverged over this time frame. And each dot represents a different virus, and you can see within like the last 5 years or a little more that there's been actually a lot of divergence that's been occurring. And the CDC's studies have said that the H3N2 viruses actually evolve more rapidly than the other influenza viruses. And so that they have needed to change the H3N2 component of the vaccine much more often than they have had to do it for H1 or Flu B even. And the other things that the CDC showed or saw that starting in 2014, there were numerous co-circulating subgroups or subclades of viruses, and one being the 3C1, a 3C2A, a 3C3A, and a 3C3B. And, and if you recall, back in the 2014-15 season, there was emergence of viruses, virus clades, that were causing vaccine failure. And those were due to the 3C2As and the 3C3As. Okay, so the drift that occurs in influenza genome doesn't only have an effect on vaccine efficacy, but it also affects other things such as laboratory tests. And it's classic that antigen assays, especially the rapid antigen assays, have had problems as the viruses have changed over the years. So whether it's, whoops, wrong button, whether it's with the established strains, and as they drift, that the antigen assays will lose sensitivity, or whether it's the new pandemic strain like they saw in 2009, that the antigen assay started to perform very poorly with those viruses. But also there's been problems with cell culture over the years, where you'll have some years that the viruses will like another cell line better than what you classically had been using, or your ability to detect the viral replication, whether by hemabsorption or by fluorescent antibodies. So what about molecular tests? In 2004, the WHO recommended targeting the M1 gene, in particular the region of the nucleotide 144 to 251, because it's considered to be a highly conserved region. But then in 2009, the CDC modified it slightly for better coverage of the pandemic H1N1 virus. And in 2008, the first FDA-approved PCR assays for flu A and B were on the market. 2009 was the first reported case of a false negative result with a commercial PCR test. And 2013 was the second case. Now, these were case reports, individual patients that had false negative results. They were unrelated, but by chance they both involved the Predictive PFlu assay, and they were on samples that were culture positive, so the virus titer was significant enough to be able to grow the sample. Although the exact primers and probes of the Predictive PFlu Plus assay is proprietary, Prodessa had indicated that there were 2 point mutations in the PFlu probe region. Then, in the winter of 2015, our laboratory we were doing an analysis. The purpose of our analysis was initially just to look at the cobas influenza A/B assay on the cobas liat. And we were going to take 275 samples, or we were taking samples for like a 2-week period. It ended up being a 15-day period, and there was 275 consecutively sent specimens to our laboratory during this time period. And the incidence of influenza was 22% in this group. And the samples were stored after the original testing, which was, depending on physician order, and about half of them were originally tested by the PFlu Plus and ProFast assays, and half by the BioFire respiratory panel, 1.7. And so then they're frozen, -80. They were thawed for testing by the cobas assay and the cobas liat, and either the respiratory panel or ProFast, depending on what test was still needed. Returned to the freezer again. And we presented this work in a poster here at CVS back in 2015. But then the next year, 2016, we tested all these samples again by the Cepheid Expert Flu. And then we published the total results. And defined a true positive as any case that was positive by 2 or more tests. And also, just to tell you some other things that were going on at this time, is that we were having some issues with PFlu, in that instead of a normal sigmoidal amplification curve of a positive result, we were seeing these curves that were funky, and they did not cross the threshold. And the technologists, they were, anytime we had a positive flu A by the Predictive assay, they were automatically reflexing to the ProFast for typing. So they went right to the typing assay when they were repeating their ProFlu assay, and the ProFast is nice and positive. And we were seeing quite a number of these. So initially, we weren't going to include ProFlu in our study because we knew something strange was going on, and we didn't want to add, muddy the waters of analyzing the cobas assay. And also at this time, that's all of a sudden the reports were coming out from the CDC about different viruses that are emerging, that are causing vaccine failure, and so we weren't sure what was going on. But it ended up being important, as we later learned and realized, and that actually with our viruses circulating in our region, that for that 2-week period of time, the ProFlu assay only had a sensitivity of about 76%. And also the expert assay had reduced sensitivity. Because we knew there's something strange with these viruses circulating, we took a lot of other viruses out of the freezer. Most of them were from a collection that we had from 2009. And so in that collection from 2009, we had 4 seasonal H1s, a few more H3s, but they're mostly all the pandemic 2009 H1N1s. And then we pulled out some flu Bs from quite a few different years. And you can see all the assays performed very well with all these different flu A viruses, and only the expert had a little bit of a problem with flu B. And this was the previous version of the expert assays, not their current. The other thing we did is that we decided to do limited detection studies on these systems with different viruses. And most of these isolates were actually from New York State Proficiency Testing Program. And so New York State, I believe they would obtain samples, clinical samples from a patient, grow them up, and use them in the proficiency testing program. And they would identify the virus type by HA, either antigenic assays or later in time sequencing. And so there's a classic Perth 16-2009-like virus, and actually ProFast and the expert had problems with this virus. There was a Texas 50 2012-like virus that was supposed to be the vaccine strain for the year. So it should have been a virus that hadn't changed too much. But actually we saw both the cobas and the ProFlu having problems with that virus in terms of a higher limit of detection. So it needs more virus to be able to detect it. So, or what tighter till you get to an endpoint, basically, right? And then there's the Swiss strain was the strain, this wasn't from the New York State Proficiency panel, but rather Jen LaPlante from New York State Department of Health provided me with this isolate, because this was this type of virus that was supposed to be the emerging virus that was causing the vaccine failure. And again, the cobas, ProFlu, and the expert had problems with these viruses. And all the assays were fine with the variant. Judy Lovchek from Indiana State Health Department provided me with a variant H3N2 isolate, and then also just using a typical pandemic H1N1 virus. All the assays worked well. And then the RP assay did have problems with the one flu B that we looked at. While we're having problems with the ProFlu assay, we provided quite a few samples to Hologic, and they did sequence and post one, the matrix gene they sequenced, and posted one to GenBank. And this is the region under the WHO target region up through to their probe, because from our studies, after the probe up to the primer wasn't having really much. They were all pretty much identical to the WHO sequences. But so this is the posted sequence, And Hologic did indicate that there was 4, even though again, their primers and probes were proprietary, there were 4 point mutations under their probe that they identified. And exactly where they are, I'm not sure, but this is just how we aligned the viruses to this Wyoming strain is the strain that's identical to the WHO sequences. So that's what I chose to align against. And you can see 4 mutations to the Wyoming strain. We sequenced 9 more of our samples, and we saw identical results. And we pulled from GenBank the Texas 50 2012 and the Swiss strain that was the emerging strain, and aligned them. And they actually had only 3 mutations, and these 2 were identical, which was kind of surprising because those 2 viruses that New York State had identified as Texas-like or Swiss-like performed differently with the assays. So we went and sequenced the Texas-like sample that we obtained from New York State. And actually that virus in the matrix gene was more like our viruses. And so, and the New York State Health Department is actually physically across the street from us. So it wouldn't be surprising that the samples that they got from our region would be somewhat similar to the rest of our viruses. And so they probably, they identified it as Texas-like from the HA gene, but it must be a chimer with a different matrix gene. The other thing is the Minnesota strain where they had, this was an H1N1 where they had a PFlu failure, and they identified 2-point mutations. It was interesting that they had I believe it was right at the nucleotide 162, where our mutations that were different from the Swiss strain was a C163T mutation. And so whether or not right in this region is the most important, it's possible. I was curious about how prevalent these mutations are in the matrix gene. So I obtained all the sequence data from the Global Initiative for the Sequencing of All Influenza Data. Their database is called PFlu. And I decided to look at 3 regions, Australia, Europe, and the US, and I got all unique H3N2 isolates that had both an HA and a matrix gene sequence available. And so Australia from 2014, since their respiratory season is during our summer, and then Europe and US just from 2014 through 15 September to April. I used Mega software for phylogenetic trees for clade determinations and also to align the matrix genes mutations. And what I saw is when I aligned the matrix genes to the WHO and CDC target regions, that there were actually 3 patterns that were present. I saw a total of 38 different patterns, but most of them were in very low numbers of virus or individual viruses that had the pattern. So there was 3 of them that were present in at least 1% of the population of viruses that I was looking at. And I also include this pattern A because this was a classic pattern that had been around for years in the matrix gene region for H3N2s. And we can see that patterns B, C, and D all co-circulated in Australia all year long for them. But that in Europe, pattern D, which has that more problematic C163T mutation, was predominant all year long. But the patterns C and D also were present all year. But in the United States, again, pattern D predominated, but actually the other patterns were only present in the fall. And then once winter came and that year's epidemic took hold, that the viruses all virtually became pattern D. And actually, in my papers, I do point out to show that there was reassortments that actually occurred between some of the different clades in the United States, so that all the different clades were acquiring this pattern D, which wasn't the case in Europe. I expanded these studies to look at a 10-plus year population, and I only looked at Europe and the US. And what I did is I took the next flu groups, so this is the next flu group, their phylogenetic tree, and I overlaid my graph on the number of mutations that have appeared per year. And you can see is that as these viruses, so this is mutations in the matrix gene, and where this is the phylogenetic tree of the hemagglutinin gene. But as you can see, the number of viruses and the diversity started to increase, so did the number of mutations in the matrix gene. And where pattern A had been prevalent for 5 years, this was replaced by pattern C for a short time, which was then replaced by pattern D. And pattern B just appeared occasionally, but in the 2013-14, it was at a more significant number. And there was a fifth pattern that was high enough numbers to make it worth noting. And this is like a pattern D, but plus a fifth mutation, a C207A, which just sits right under the CDC probe binding region. And whether it's significant or not, I can just tell you that Marie Landry at Yale has been using for years the CDC protocol, and she has had more recently problems with detecting H3N2s. So it could possibly be a significant mutation for the performance of assays. But luckily that pattern has also been disappearing. So because of these changes, commercial manufacturers are required to monitor their tests for mutations against the circulating influenza strains. And because of it, 3 manufacturers have modified their assays to address these concerns, or part of the reasons why they've made the changes. And so the Cepheid Expert Express Flu and Expert Flu RSV XC assays have expanded their target region, and the BioFire Film Array, Respiratory Panel 2, and the Hologic Panther Fusion Flu A B RSV assay. And so we were doing an analysis on the Panther Fusion and particularly the Flu A B RSV assay. But as I mentioned, this is Hologic has chosen to use a modular syndromic approach so that you can choose the tests that are right for the patient population. And so they have a Flu A B RSV, a adenovirus metapneumo rhinovirus assay, and then a paraflu assay. But I'm just going to be focusing on the Fusion Flu A B RSV assay and with its influenza redundancy, because what they've actually done is they designed their assay so it has amplifies 2 target regions and uses multiple probes to capture more strains of virus. And so to do this study, I took the same samples that we have tested before in the other studies. So the 275 consecutive NP samples from 2015, as well as many of the other additional viruses. There was a few that there was no longer sample available. And to let you know, this was really going to be a challenge for the fusion assay, because these samples now have been frozen and thawed at least 4 times, and some of them even more if they were involved in a discrepancy analysis in the past. We also, because our goal is to be looking at the other fusion assays, respiratory assays, we pulled quite a few other samples positive for other viruses. But I'm just using the data of them being influenza negative to add to the samples. And what we saw is the fusion was able to meet the challenge for these compromised specimens, highly compromised specimens. In all 61 samples, that were considered true positives in our previous studies were still positive with the fusion assay. In addition, there were 4 other samples that were considered previously in our previous analysis considered to be false positive by either ProFast or by the COBAS assay because they were only positive by those assays, but the fusion was positive with those as well. And then there was an additional 10 samples that were positive by fusion only. And so we tried to do a discrepancy analysis, but a lot of these samples just had a very low volume, but we were able to retest them with the ProFast assay using double the amount of volume of sample that you normally would test and extracting them on the EasyMag. And we were able to get an additional 4 positive samples out of there. So this changed our true positive number of true positives so that there's actually 8 more true positives than we previously identified. And so this changes these statistics of our previous studies, and that the incidence of H3N2 during that 2-week time period was actually 25%, not 23%, and the sensitivity of all the other assays are actually much lower, and where the fusion assay's sensitivity was 100%. Looking at all the other viruses in an aggregate of the sensitivities of all the different flu strains, we could see that fusion sensitivity was 100% for the H1N1s, the H3N2s, and flu Bs, where that was not necessarily true with all the other assays. Okay, so what about the samples, the additional samples that were positive by fusion only? There was actually, in the additional samples that we tested, there was one more that was positive for Flu A that was not positive by any other assay. But the only positive samples by fusion that were negative by all the other assays were only samples that appeared during periods of high influenza prevalence. And when there was, like in the samples that were from the summertime, there was never a false positive, or even other seasons, especially during H1N1 seasons. And so this one additional sample was 2014, and all of a sudden, April 2014, we were experiencing a surge. It was an H1N1 year, but we were suddenly experiencing a surge of H3N2s. And so the 6% was probably more like 10%, but we had not recognized at that point in time that we were missing some cases. And so 40% of the viruses should have had the problematic sequences. We also did the limited detection study. And this is truly amazing because look how much lower the limit of detection of the fusion assay compared to all the other assays. And actually the limited detection is on the order of 0.2 to more than 3 logs lower with fusion than it is with the other assays. So, you know, in my mind, that this system can detect so little virus that it's going to be nearly impossible to confirm. There's no other method to confirm these extra positives because nothing can get to that low. So in conclusion, there's significant drift has been occurring in H3 strains in particular, and that this drift has affected the performance of many molecular tests in addition to vaccine efficacy. Panther Fusion's assay design has influenza redundancy to safeguard against viral drift. And so it uses, again, 2 targets with multiple probes. And the exquisite sensitivity was demonstrated with both our clinical specimens and our limit detection studies. And the fact that the unverified additional positive samples were only observed during a period of elevated H3N2 prevalence and the such low level of virus that this assay can detect makes me feel that these positives are highly likely to be true positives. So it begs the question is whether or not the Panther Fusion is the next gold standard or the new gold standard for Flu A/B testing.